Photosynthesis takes place in an organelle in a plant cell called the chloroplast. Within the chloroplast, there are membranous sacs called thylakoids, which hold chlorophyll. The outside of the thylakoids are called the stroma, while the inside is called the lumen. The membrane of the thylakoid itself is a phospholipid bilayer. The outside of the thylakoids are called the stroma, while the inside is called the lumen. The membrane of the thylakoid itself is a phospholipid bilayer. The first step in the light-dependent reaction chain is when a photon from the sun strikes photosystem II. This causes photosystem II to become excited and release an energized electron, which is carried by a protein called the mobile electron carrier to the cytochromatic complex, which uses the energy to bring in protons of hydrogen into the lumen. Meanwhile, back at photosystem II, to compensate for the loss of an electron, it splits a water molecule to gain an electron and releases oxygen into the air. As the electron carrier continues from the cytochromatic complex to photosystem I, which re-energizes the electron. The electron then moves to NADPH reductase to be used in the creation of NADPH. As that is all happening, protons have been added into the lumen by splitting water or from the cytochromatic complex. This creates an imbalance which forces the protons to leave the lumen through a protein called ATP synthase. It takes ADP and combines it with a proton to create ATP. The second part of photosynthesis is the dark reactions, or the light-independent reactions. This is also called the Calvin cycle. It takes place in the stroma of the chloroplast. There are 3 stages of the Calvin cycle: carbon fixation, reduction, and regeneration. The first step in carbon fixation is where CO2 is fixed by adding it to a 5-carbon compound, RUBP. This creates a 6-carbon compound, which is unstable and falls into 2 3-carbon compounds. This is the first stable product of the cycle. Reduction is the second step and turns the 3-carbon compounds from a low energy state to a high energy state using ATP. Then NADPH reduces the compounds into G3P. At this point, some G3P leave the cycle to create glucose. The remaining move to the regeneration step. The final step is regeneration. It takes the remaining G3P, and with the energy of more ATP, it makes the original 6 RUBP 5-carbon compounds. That is the Calvin cycle.
Cellular respiration can be divided into 3 different stages: glycolysis, Krebs cycle, and electron transport. Starting with glycolysis, the main idea is to transform glucose, which is a 6-carbon molecule, into a useful form of energy, known as ATP. Although this may seem quite simple, the actual work involved is quite complex. In order to completely convert/create ATP, glucose must first be transformed into 2 pyruvates, which are 2 3-carbon molecules. In order for this process to occur, a small amount of ATP is already needed to enact this. You basically have to give so you can get. 2 ATPs are needed. Throughout this process, glycolysis allows for the result of 2 pyruvates along with 4 ATPs and 4 NADH, NADH being an energy-rich molecule. The second stage of cellular respiration is known as the Krebs cycle. This process takes place in the inner membrane of the mitochondria. This process takes the products produced in glycolysis and creates another pair of ATP. This all occurs by oxidation. This basically means that the pyruvate's carbon molecule interacts with oxygen, causing them to use an electron. This is the creation of acetyl, coenzyme A, and carbon dioxide. That's when a hydrogen interacts with an NAD and creates NADH. That makes the useful amount of products that can be used in the next step of cellular respiration. The final stage of cellular respiration is the electron transport chain, leading to the forming of ATP in the inner membrane of the mitochondria, which make up 36 ATPs. All the electrons in the Krebs cycle will provide the energy that will work as a pump along a chain of channel proteins across the inner membrane of the mitochondria, where the Krebs cycle occurs. These proteins will swap these electrons and protons inside the center of the mitochondria across its inner membrane, but once they're out, the protons want to get back in to the other side of the membrane because there's a lot of other protons up there. As we learned, nature always tends to seek balance on the other side of the membrane. So all these protons are allowed back in through ATP synthase.